So imagine a world map for a second. You've got the polar bears up at the freezing North Pole, and then you have the dense jungle right at the equator.
Right. Totally different environments.
Exactly. And by the basic laws of geography, they never cross paths, right? They exist in completely different, highly compartmentalized zones. But today, we are going to look at a biological anomaly where the jungle has somehow hardwired itself directly into the ice caps.
It is a wild concept to picture.
It really is. Welcome to the Deep Dive, everyone. We have a massive stack of recent genomic research to unpack today, specifically centered around a landmark 2026 Nature paper. The title is Centromeric Footprints Preserve Telomere Integrity in ALT Cancers. And basically, scientists have discovered a cellular crime scene.
A crime scene where the fundamental laws of biology are just being completely broken.
Yeah, exactly. Our mission today is to figure out how, in about 5 to 10% of all cancers, the absolute middle of the chromosome has physically inserted itself into the extreme ends.
Your world map analogy actually captures the scale of this anomaly perfectly, because in the landscape of the human genome, compartmentalization isn't just about keeping things neat and organized. I mean, it is an absolute requirement for preserving genetic integrity.
Right, you can't just mix and match these zones.
Exactly. Aside from incredibly rare, highly specialized exceptions like during meiosis and certain types of yeast, the middle and the ends of your chromosomes simply do not mix. So the core mystery we are dissecting today is how these two distinct domains physically touch, how they fuse, and most critically, why a cancer cell intentionally does this to survive.
Okay, let's unpack this. If you were listening to this deep dive, you probably already know the basic architecture we're dealing with here. The poles in our map are the telomeres, right? The extreme ends of the DNA strand.
Yes, the physical ends.
And the equator is the centromere, that massive structural landmark right in the middle. We're used to thinking of telomeres as the cell's physical crumple zone. They have that repeating DNA sequence, TTKG, that sacrifices itself.
Right. It's like the plastic aglets at the ends of your shoelaces.
Yes, exactly. It stops the DNA from fraying. It shortens a little bit every single time the cell divides, which protects the crucial genes that are located further inward.
Precisely. And then, on the other hand, the centromere serves a completely different, purely structural purpose. It is the anchor point.
The anchor point for cell division.
Right. Its unique DNA code is made of these really long, higher-order repeat arrays of what we call alpha satellite DNA. And this DNA contains specific CENP-A box motifs. And the whole point of this code is to build the kinetochore.
Which is the hook.
Exactly. It's the physical hook. During cell division, cellular cables attach to the kinetochore to pull the duplicated chromosomes apart into two brand new cells. So you have one region that's a buffer and another that is a heavy-duty structural anchor.
Which brings us to the anomaly, right? ALT cancers. Because most cancers stay immortal by hijacking an enzyme called telomerase. They just endlessly rebuild their telomere crumple zones so they never age.
But about 5 to 10% of cancers don't use telomerase at all.
Yeah, and that is wild to me. They rely on this rogue, super aggressive survival mechanism called Alternative Lengthening of Telomeres, or ALT.
Yes, ALT.
So let me just give a quick recap for you listening before we move into the mechanics. Number 1, telomeres are the ends, made of TTKG. Number 2, centromeres are the middle anchor point made of alpha satellite DNA. And number 3, ALT cancers don't use standard telomerase.
That sums up the playing field.
So here is the question we need to answer next. If you are an ALT cancer cell and your telomeres are degrading to the point of catastrophic cell death, but you don't have the normal enzyme to rebuild them, where do you steal the replacement code from?
You steal it from the sturdiest structure available. You steal it from your own center.
Wow.
Yeah. Researchers started noticing this happening in ALT cancer cells, specifically in these heavily studied lab cell lines, like U2OS cells. They saw that the telomeres were physically clustering together into these dense hubs.
Right, the APBs, ALT-associated PML body.
Exactly. But actually seeing that happening is, it's a massive technical challenge.
Oh, I bet. I mean, we're talking about regions of the genome that are notoriously repetitive, tightly packed inside a microscopic nucleus. You can't just peer through a standard high school microscope and watch the DNA mingle.
No, standard microscopy wouldn't even scratch the surface here. They had to deploy something called PNA-ish.
Okay, PNA-VESH-H. Break that down for us.
It stands for peptide nucleic acid fluorescence in situ hybridization.
That is a mouthful.
It is, but it's an incredible tool. The brilliance of it isn't just that it uses glowing, color-coded molecular probes. It's the fact that it uses synthetic peptide nucleic acids.
Oh, so it's not normal DNA.
Right. Normal DNA has a negative charge, which creates electrostatic repulsion when things get crowded. Because these synthetic probes lack that negative charge, they don't repel each other. They can bind their target sequences, like the centromeric alpha satellite repeats and the telomeres, with just incredible affinity and precision.
So they basically lit up the centromeres with one glowing color and the telomeres with another color, and just looked for where the colors smashed together.
In a sense, yes. And when they looked at normal non-ALT cells like standard HeLa cells, the colors remained strictly separated. The poles and the equators stayed in their designated lanes.
But in the ALT cells?
In the U2OS ALT cells, or in cells where they artificially force the ALT mechanism by deleting a regulatory protein called ATRX, the probes perfectly overlapped. The colors merged.
But how do they know it's not just an optical illusion? Like, if you look at it from a top-down view, one might just be floating above the other.
Right? Exactly. You have to rule that out. So to prove they were actually touching, they used SIM, structured illumination microscopy.
Oh, right. This is the one that overcomes the normal diffraction limits of light.
Precisely. SIM projects these really intricate grid patterns of light over the sample. It allows scientists to reconstruct an image at super resolution. And that definitively confirmed that these two regions were in direct physical contact.
Okay, but I want to clarify something for the listener. This isn't just passive floating around. They aren't just accidentally bumping into each other in a crowded nucleus. This is a highly active, chaotic construction site.
It's an incredibly aggressive exchange of material. And to capture that action, the researchers used two more assays, exoFISH and EDU incorporation.
Okay, what do those do?
Well, exoFISH is crucial because it detects actual physical breaks in the DNA strands inside cells that are just going about their normal cycle completely unperturbed. And then EDU incorporation acts as a tracer.
A tracer for what?
For new DNA. It leaves a permanent, visible marker wherever brand new DNA is actively being synthesized. And what they found was a massive surge in EDU incorporation precisely at the spots where the telomeres and centromeres were overlapping.
Oh, wow. And the timing of this is what really stands out to me. Because this active DNA synthesis peaked during the G2 phase of the cell cycle.
Yes, the timing is a massive clue.
For those keeping track, the G2 phase is the exact moment when the cell has finished copying its DNA and is doing a final check before dividing. It's the moment when the ALT mechanism goes into absolute overdrive. All the core molecular mediators, proteins like PML, BLM, and PLLD3, they're just swarming the area.
Right, and the technical term for what they're doing is break-induced replication.
Okay, explain break-induced replication.
It's basically a desperate repair mechanism. When a DNA replication fork collapses or breaks, the cell panics. It tries to restart the process by invading a neighboring similar sequence, and it uses that neighbor as a template to keep copying.
So the cancer cell is leveraging this panic response.
Exactly. It uses the structural breaks at its degrading telomeres to literally launch an invasion into the centromeric templates.
It's like, imagine a failing bridge. The edges are crumbling into the river, so the construction crew starts violently ripping rebar and concrete out of the central load-bearing pillar and haphazardly welding it to the crumbling edges just to keep the bridge open for one more day.
That is a very accurate, albeit terrifying, way to picture it.
So let me recap this section really quickly. 1. Telomeres and centromeres are physically touching in ALT cells. 2. Glowing probes and super-resolution microscopes prove it. And 3. They are actively copying DNA from each other during the G2 phase using break-induced replication.
Exactly.
Which brings up an obvious question for you listening. If the cell is violently ripping code from the middle and welding it to the ends, what does the permanent scar look like in the DNA? What's left behind?
To find that scar, the researchers had to move away from imaging and transition into deep sequencing, but you immediately hit a wall with traditional sequencing when it comes to telomeres.
Because traditional sequencing, like Illumina short-read sequencing, basically shreds the genome into tiny fragments, right?
Right. It shreds it into tiny pieces, and then computers try to overlap them to rebuild the code. But because the telomere is just TTTage repeated thousands of times, trying to piece those short reads back together is like trying to assemble a massive jigsaw puzzle, where every single piece is the exact same shade of blue.
Well, that makes sense. You have absolutely no idea where you are in the sequence. You're just looking at blue sky.
Exactly. So to get around this, they used an advanced method called telosec.
Telosec. How is that different?
It's an absolute game changer. It leverages long-read nanopore technology. So instead of shredding the DNA into tiny bits, telosec pulls ultra-long, continuous single molecules of DNA through a microscopic protein pore.
Really pulling the strand through a hole.
Yes, and as the DNA passes through, the machine reads the specific electrical current changes for each base pair. This allows scientists to map the exact coordinates of those long, repetitive ends without losing their place.
That is incredible. And so when they ran telosec on normal cells or cancers that rely on standard telomerase, they just saw clean sequences, right? Just standard blue puzzle pieces.
Clean sequences, entirely normal telomere repeats. But then they looked at the ALT-positive cancer cell lines like G292, SOUS2, and those U2OS cells we talked about earlier.
And that's where the scar appeared.
That's where they found it. Between 1 and 5% of the long telomere sequencing reads in those ALT cancers suddenly contained 17 base pair motifs of alpha satellite DNA.
The centromere code.
The centromere code was physically embedded right inside the telomere code.
And it wasn't just scattered randomly either, was it?
No, the mapping was incredibly specific. Over 95% of these alpha satellite insertions were wedged exactly at the subtelomere-telomere boundary.
Right at the biological root of the telomere.
Exactly. We're talking about near-identical insertions of long strands of centromeric code, complete with those CENP-B boxes meant for anchoring wedged directly into the genome's crumple zone.
But here's the part that really grounds this entire phenomenon and makes it so important for you listening. This is not just some weird artifact of lab-grown cell lines mutating in a petri dish.
No, that's a vital point. The researchers investigated primary pediatric neuroblastoma tumors.
Real human pediatric cancers.
Real clinical samples from young patients. And the clinical analysis revealed that the ALT-positive neuroblastomas contained approximately 3 times more of these chimeric sequences, these hybrid alpha satellite and telomere mutations, compared to the ALT-negative tumors.
3 times more. So stepping back for another quick recap. 1, telosec finds the actual physical scar at the DNA level. 2, those centromere alpha satellite sequences are wedged right where the telomere starts. And 3, this exact mutation is actively happening in real human pediatric cancers.
It's a verified clinical reality.
Okay, but here's a huge question. DNA sequences don't just exist in a vacuum. If you fundamentally change the sequence, you fundamentally change the physical packaging of the chromosome, right? The hardware has to react to the new software. So what happens to the protective packaging of the DNA?
That introduces a critical new player, CENP-A.
Okay, CENP-A. What is that?
So your DNA is tightly spooled around protein complexes called histones. It keeps everything organized. CENP-A is a highly specialized histone variant. You can think of it as a master molecular ID badge.
An ID badge for the centromere.
Yes. When CENP-A is deposited onto a section of DNA, it basically signals the cell's structural machinery, saying, hey, this is the spot. Build a functioning centromere exactly here.
So if the centromere code is suddenly sitting at the telomere, does the ID badge go there too?
To find out, the researchers used CUN analysis.
CUN, another great acronym.
Right. CUN acts like a highly precise molecular GPS. It maps exactly where specific proteins are binding to the DNA. And they combine this with SNP tagging, which is a fluorescent tracking method.
So they made the proteins glow so they could follow them.
Exactly. And they discovered that CENP-A, the master builder for the centromere, was indeed physically stationed at the telomeres of these ALT cells.
Okay, but wait. Normal telomeres are heavily guarded.
Okay.
I mean, it's locked down by epigenetics. Precisely so, stray machinery doesn't accidentally build weird structures on the delicate ends of the chromosomes. How does CENP-A just waltz past the security?
That's where the researchers had to utilize an incredibly sophisticated tool called demilosec.
Demilosec.
It stands for directed methylation with long-read sequencing. And what makes it so brilliant is that it reads protein binding and DNA methylation simultaneously on the exact same strand of single molecule DNA.
Here's where it gets really interesting. Because methylation, for those who might need a refresher, methylation refers to the chemical locks that are added to the DNA backbone to shut down access to certain genes.
Precisely. Now, usually, to see both protein binding and methylation, you have to run 2 separate assays and try to mathematically overlay the data, which is messy. But demilosec uses an antibody to target your protein of interest, which in this case is CENP-A.
Okay.
Attached to that antibody is a special enzyme. Wherever the antibody finds CENP-A, the enzyme artificially methylates the DNA right next to it. It essentially stamps a footprint into the DNA.
Oh, that's clever.
Very. Then, the long-read sequencing processes the single DNA molecule, reading both the cell's natural methylation locks, specifically MCG methylation, and the artificially stamped CENP-A footprints at the exact same time.
Wow. And what did they find when they looked at the locks and the footprints together?
The data showed an undeniable inverse relationship. They found that the CENP-A footprints appeared exactly and only in the locations where the natural DNA methylation locks had vanished.
The locks were gone.
Completely. The telomeres on chromosomes 1, 11, and 14 were hypomethylated, meaning their locks were stripped away and CENP-A had moved right in. But on chromosomes 5, 20, 16, and 17, the methylation locks were still securely in place.
And CENP-A was blocked out of those ones.
Completely blocked out.
Okay, I have to push back here, though. We see this all the time in biology. Correlation isn't causation. Just because the epigenetic locks are missing in the exact same spots where CENP-A has invaded, that doesn't absolutely prove that the missing locks caused the invasion. Maybe some other rogue mechanism ripped the locks off and dragged CENP-A in with it?
You're absolutely right to question that, and the researchers anticipated that exact skepticism. So to prove causation, they designed a targeted smoking gun experiment. They used a fusion protein called dCas9-TET1-CD.
Okay, break that down for me. I know Cas9 from CRISPR gene editing. It's usually the genetic scissors that cut DNA.
Right, but the D in dCas9 stands for catalytically dead. It's a broken pair of scissors. It doesn't cut the DNA at all. It just acts as a programmable molecular GPS. It navigates to a specific programmed address, in this case, the telomere.
Oh, I see. So it's just a delivery vehicle. What is it delivering?
It's tethered to TET1-CD, which acts as a molecular lockpicker. Its sole job is to actively strip away methylation.
So they literally sent a lockpicker directly to the telomeres to force the locks open and see what would happen.
Yes. By artificially targeting demethylation strictly at the telomeres, they observed that the endogenous levels of CENP-A at those telomeres surged by 1.5-fold.
Wow.
This definitive experiment proved that the epigenetic barrier is the primary gatekeeper. If you strip away the methylation, the centromere machinery naturally floods into the telomere.
Okay, time for another quick recap. 1, CENP-A is the centromere builder ID badge. 2, it snuck into the telomere because the epigenetic locks, the methylation, were removed. And 3, forced unlocking proves that this removal is what allows CENP-A to enter.
Spot on.
But if you are listening to this, you are probably thinking the exact same thing I'm thinking right now. This sounds totally suicidal for the cell.
It does seem counterintuitive.
Let's think about the mechanical reality of cell division. You said earlier that CENP-A is the signal to build the kinetochore, the hook that grabs the cellular cables to pull chromosomes apart into two new cells. If you build an anchor point that is meant to be violently ripped apart at the absolute extreme end of a fragile chromosome arm, won't the cell just grab that end and tear the chromosome to shreds?
It is the most critical functional question in this entire deep dive. Because you're right, the mechanics of division are completely unforgiving. Under normal circumstances, a deposition factor called HJURP places the CENP-A badge.
Okay, HJURP.
Right. And then that badge recruits two other proteins, CENPC and NDC80, which form the actual physical hooks that engage the pulling cables, the microtubules. If a fully functional mature kinetochore forms at the telomere, the mechanical tension during division would be catastrophic. The chromosome arm would simply snap.
And when a chromosome snaps, the fragmented pieces get left behind, forming what biologists call micronuclei, right?
Yes.
And a cell that is filling up with micronuclei is experiencing massive genetic instability. It triggers internal alarms that almost always end in the cell killing itself.
So to see if this was happening, the researchers ran a targeted micronuclei assay.
What did they look at?
They compared chromosome 11, which, as the Dymalosex showed, had lost its locks and gained the CENP-A footprint against chromosome 4, which had maintained its locks and had no CENP-A at its telomere.
Okay, so the ultimate test. Does chromosome 11 tear apart?
If the structure at the end of chromosome 11 was a fully active centromere, it should be tearing apart constantly. But the assay revealed that chromosome 11 did not missegregate, nor did it form micronuclei any more frequently than chromosome 4.
Wait, really? It builds the foundation for the anchor, but it somehow avoids the mechanical tearing. How does it survive that tension?
By not finishing the construction.
Oh.
Yeah. And the researchers wanted to prove this by seeing what would happen if the construction was actually finished. So they engineered a forced failure scenario.
How do they do that?
They took that deposition factor we mentioned, HJURP, and physically fused it to a protein called TRF1, which naturally binds to telomeres. This forced the cell to massively over-recruit HJURP directly to the chromosomal ends. It artificially drove the pseudocentromere to fully mature.
They forced the hook to finish building. And what happened?
It was devastating for the cell. When they forced the issue, it caused massive missegregation and widespread micronucleation. The chromosomes were shredded, exactly as you hypothesized.
Which means, so what does this all mean? The cancer is pulling off an incredibly delicate, honestly terrifying balancing act. It's what we could call a Goldilocks kinetochore.
A Goldilocks kinetochore is the perfect way to describe it.
The cancer cell allows just enough centromeric material and CENP-A to assemble at the telomere. This partial structure acts like a rigid splint, patching up the degrading crumple zone and saving the cell from immediate telomere collapse. But the cancer strictly limits the abundance of these proteins.
Yes, it holds the construction back.
It holds it back so it never matures into a fully functional hook.
Yeah.
If it grew too strong, the microtubules would attach and the cell would tear itself apart during division. It is surviving purely by walking a tightrope of structural instability.
The adaptability of tumor biology is genuinely astounding. To synthesize what we've covered today, the cancer is exploiting break-induced replication to literally copy-paste the structural integrity of its own center to patch up its fraying edges. It accomplishes this by selectively manipulating its own epigenetic locks, the methylation, to allow the master builder CENP-A to enter the telomere. But, as you said, it tightly regulates the process to avoid catastrophic mechanical failure.
It's brilliant and horrifying all at once.
Truly, and it's really worth highlighting that none of this granular understanding would be possible without the incredible leap in modern tools like Telesec and Demelisec, which allow us to finally read these heavily disguised, highly repetitive regions of the genome.
It really redefines how we view the resourcefulness of a tumor. The fact that it can read its own pathological instruction manual and rewrite the rules of geography to stay alive.
It changes the paradigm.
It does. And as we wrap up, I want to leave you, our listener, with a final thought to mull over. Something that builds on everything we just talked about. We've just established that this highly aggressive subset of ALT cancers relies entirely on this perfectly balanced Goldilocks pseudocentromere, right?
Right, it needs that precise balance.
It needs the splint to survive, but it completely relies on that splint remaining incomplete so it doesn't tear itself apart.
Yeah.
So what happens if future targeted therapies figure out how to hijack that balance?
Oh, that's a fascinating angle.
Right. What if a future drug doesn't try to destroy the cancer's telomere, but instead artificially pushes that pseudocentromere to full maturity? Could we essentially trick the cancer cells into finishing the construction, forcing them to literally tear their own chromosomes apart the next time they try to divide?
Using their own survival mechanism as a weapon against them.
Exactly. It exposes a completely new mechanical vulnerability that we didn't even know existed until this paper. So keep questioning the microscopic mechanics in the world around you, and thanks for joining us on this deep dive.